microscope versions where you can see the endoplasmic reticulum dotted with ribosomes. You can see the outside versus the inside of the cell and the bilayer, the phospholipid bilayer, also known as the cell membrane. So here's a plant cell. So the difference are a central vacuole, animal cells do have vacuoles, they're just small. Plant cells always have a central vacuole so that they can keep water in it and remain strong. They have a cell wall. So if you see a eukaryote with a cell wall, it's likely a plant. And then everything else is pretty much the same, except for these. These are pores through the cell wall. And those are pretty much the differences with plants. So these can go linked to other pores in an adjacent cell wall, which is what you're seeing here. Here's one cell wall, and here's another cell wall, and here's another cell wall. So these actually go through both cell walls. But plants do still have a cell membrane inside of their cell wall. So here's a scanning electron microscope of a Golgi apparatus. It kind of looks like stacked pancakes. And then here's a chloroplast. It has a lot of little stacks in it. So each of these little guys is just like a stack, which is used to absorb light. So ribosomes translate the nucleotide sequence of a messenger RNA molecule into a polypeptide, aka primary structure. Of a protein. They occur in both prokaryotic and eukaryotic cells and consist of one large and one small subunit. So there's the large subunit and the small subunit. Part of the ribosome, which is made up of ribosomal RNA, which we see that here, which is also shortened to rRNA. And there's small protein molecules that it's bound to. It works as an enzyme. To catalyze peptide bonds. So it can also be referred to as a ribozyme because it's a ribosome and an enzyme. Ribosomes are not membrane-bound organelles, so not membrane-bound organelles, which is why they can be in prokaryotes and eukaryotes. In eukaryotes, they are free in the cytoplasm. They're usually included in lists that ask for functions of organelles, but they are not actually an organelle. In eukaryotes, they are free in the cytoplasm or attached to the endoplasmic reticulum or inside the mitochondria and chloroplasts because they make their own proteins. In prokaryotes, ribosomes float freely in the cytoplasm because there's nothing for them to attach to. The nucleus is usually the largest organelle. It's the location of DNA and DNA replication. It's the site where DNA is transcribed to RNA. It contains the nucleolus, where assembly of ribosomes from RNA and proteins begins. So here we have our bilayer around our nucleus, and then in there is the nucleolus, which is usually just a dark spot in the nucleus, and then we have our DNA all around it in its unwound form here. So the nucleus is surrounded by two membranes that form the nuclear envelope. Nuclear pores in the envelope control movement of molecules between the nucleus and the cytoplasm. So nuclear pores are just holes in the nuclear envelope, or the membrane around the nucleus. In the nucleus, DNA combines with proteins to form chromatin in long, thin threads called chromosomes. So chromatin is just the genetic material, and then when it winds up, it is a chromosome. And that's generally what a chromosome looks like. A duplicated chromosome is that common X formation that you see a lot. This is duplicated. This is a single chromosome. The outer membrane of the envelope is continuous with the endoplasmic reticulum. So it just kind of comes off of it into the endoplasmic reticulum around it. The endomembrane system includes the nuclear envelope, endoplasmic reticulum, Golgi apparatus, and lysosomes. And these are just membranes that can bubble off of and fuse with other membranes. So this one's bubbled off, and then it can go fuse by joining the other membrane and breaking those points to fuse with it, which is how things can be pushed out or into a membrane. Tiny membrane surrounded vesicles shuttle substances, so this is a vesicle here, between the various components as well as to the cell membrane. So here we have our nucleus, and here it is continuous with the endoplasmic reticulum, so the nuclear envelope is pretty much the same as the start of the endoplasmic reticulum here. And then here are vesicles that come off the endoplasmic reticulum and go into the Golgi apparatus. So the cis region is towards the nucleus, and then the trans equals away from the nucleus. So things can go either way. They can come from the cis region and go towards the trans region, getting sorted and put outside of the cell, or they can be taken in from the outside and go towards the cis region and get put into the endoplasmic reticulum. The endoplasmic reticulum, also shortened to ER, is a network of interconnected membranes in the cytoplasm with a large surface area. There are two types of endoplasmic reticulum. There's the rough endoplasmic reticulum, so that's with ribosomes, and the smooth endoplasmic reticulum, that's without ribosomes. So this is where proteins can be made, and proteins cannot be made on the smooth endoplasmic reticulum. Rough endoplasmic reticulum has ribosomes attached to its outer surface. Newly made proteins enter the rough endoplasmic reticular lumen, which is just the inside, where they are chemically modified and tagged for delivery to specific locations. The proteins are transported in vesicles that pinch off from the endoplasmic reticulum. All secreted proteins and most membrane proteins pass through the rough endoplasmic reticulum. Polypeptides are transported into the rough endoplasmic reticulum lumen as they are being synthesized. So you have your ribosome, it's on the surface of the endoplasmic reticulum. What's going on? Okay. So as it's synthesized, you have your mRNA here, and then your protein gets made into the lumen. Here's your polypeptide. It's supposed to be a dash because it's one word. Okay. In the lumen, they are folded into their tertiary structures. Many are linked to carbohydrate groups, becoming glycoproteins. Many glycoproteins are important in recognition and interaction between cells. So a glycoprotein is just a protein with a carbohydrate coming off of it, which usually look like branched structures, almost tree-like. So this is a glycoprotein. I'm always running out of room for my words. Many glycoproteins are important in recognition and interactions between cells. The smooth endoplasmic reticulum, SER, is more tubular and no ribosomes. So it's just kind of like a tube that's interconnected. With its double membrane system. Okay, so it chemically modifies small proteins such as drugs and pesticides. Since it's the site of glycogen degradation in animal cells, it's the site of synthesis of lipids and steroids. It stores calcium ions, which trigger many cell responses. So those are its functions. It chemically modifies small molecules. It's the site of collagen degradation. It's where lipids and steroids are synthesized, and it stores calcium ions. So it has chemical modification, degradation, and synthesis, as well as storage functions. The Golgi apparatus is flattened sacs called cisternae and small membrane enclosed vesicles. So it's just an apparatus of all these things combined. It receives proteins from the rough endoplasmic reticulum and can further modify them. It concentrates, packages, and sorts proteins. It adds carbohydrates to proteins. It's the site of polysaccharide synthesis for plant cell walls. So it's really kind of like a post office warehouse. It says, okay, here's a whole bunch of packages. Dark ones go over here. And light ones go over here. Or whatever it uses normally. certain molecular groups on the proteins to sort them. So it sorts them off into different vesicles so they can go where they need to go. The Golgi apparatus has 3 regions. The cis region, which is towards the nucleus, it receives vesicles containing proteins from the endoplasmic reticulum. The trans region, which is away from the nucleus, it's where vesicles bud off from the Golgi apparatus and travel to the cell membrane or to lysosomes. And then there's the medial region, which is in between the trans and cis regions. So you have your stacked pancakes, and those are the medial. And then you have your vesicles that are going off towards the nucleus. That's your cis. And then you have your vesicles that are pinching off towards the cell membrane. And that's the trans region. So here's a better picture of that. Nucleus, cis region, medial region, and then trans region. And then the lumen is inside of it. So these can either go for use inside the cell or to be released. Macromolecules may enter the cell by phagocytosis. Part of the cell membrane encloses the material and a phagosome is formed. Phagosomes then fuse with primary lysosomes to form secondary lysosomes. Enzymes in the secondary lysosome hydrolyze the food molecules. So here you have your cell membrane and a little bubble can form around a certain material. There's usually like sensors or something that helps it know that the stuff it wants is in there, or it just might be trying to take in random stuff from the environment. So this is phagocytosis. So it's how things can come into the cell. This pinches off and you have a bubble vesicle that has the stuff it wants in it, and the membrane closes up behind it. So these can go then fuse with the lysosome. They do this little fusion thing. Which has molecules that can break down those particles and help them hydrolyze the food molecules. So here we have the inside of the cell, the Golgi apparatus, a primary lysosome which has those enzymes that can break down food. So here it's taking in food. It's a phagosome. It merges with a secondary lysosome. It breaks down the food. The good things get taken into wherever they need to go, and then the waste products get excreted from the cell. Here's a scanning electron microscope of it actually happening. Here's a food particle being taken in. Here's a phagosome. Here's the lysosome. Here's what it looks like when it's a secondary lysosome. And then food can be excreted as well once it's done. So phagocytes are cells specialized to take in material and break them down. Autophagy is the programmed destruction of cell components. cells break down their own materials and even entire organelles within lysosomes. Like if they have too many or something, that's autophagy. Lysosomal storage. Diseases occur when lysosomes fail to digest cell components. So lysosomes are just an organelle for degradation. I don't know if that was on our earlier diagram, so I'm just putting it there. In eukaryotes, breakdown of energy-rich molecules begins in the cytosol. The partially digested molecules enter the mitochondria, where chemical energy is converted to energy-rich ATP. Cells that require a lot of energy often have more mitochondria. So your muscle cells will have a lot more mitochondria than your intestinal cells. So mitochondria have two membranes, an outer membrane, which is very porous, and an inner membrane that has extensive folds. And apparently it just glitched. Oh, you get the idea. These are called cristae. The fluid-filled matrix contains enzymes, DNA, and ribosomes. So we'll go into more mitochondrial structure and function later when we do metabolism. And ATP synthesis. So plant and algae cells contain plastids that can differentiate into organelles. Some are used for storage. The chloroplast contains chlorophyll. It's the site of photosynthesis. So the chloroplast has a whole bunch of stacks in it, which have chlorophyll in them, along with some other pigments, which we will learn about in our plant pigmentation lab. So there's chlorophyll A, chlorophyll B, carotenoids, and xanthophyll. And these all work together to absorb the wavelengths of light and make sugar out of light waves. So photosynthesis converts light energy into chemical energy. This is an anabolic process. Anabolic means building. Catabolic is degradation. Metabolic is both. So anabolic is the building processes, catabolic is the degradation processes, and metabolic processes are both of those combined. So photosynthesis equation is energy in the form of light plus carbon dioxide yields sugar, water, and oxygen. So super useful for us humans who super need sugar, water, and oxygen for our catabolic processes. So chloroplasts have two membranes, plus internal membranes called thylakoids. Thylakoids. Granum is a stack of thylakoids. So when I was drawing that chloroplast, when I was doing these are the thylakoids. Let's draw an arrow to that word. And a granum is the whole thing. It's a stack of thylakoids. Light energy is converted to chemical energy on these membranes. A stroma is an aqueous matrix around the grana containing ribosomes and DNA. So if we go back, this stuff right here and around here, that's the stroma. Carbohydrates are synthesized in that. So other plastids, chromoplastids make and store red, yellow, and orange pigments, especially in flowers and fruits. So chromoplasts just contain pigments and they give flowers and fruits their color. So it's just a membrane-bound organelle with a whole bunch of little pigment molecules in it to give it that color. So leucoplasts store macromolecules such as starch. So this is just another type of plastic. So this happens a lot in potatoes and beets and carrots. So these are your root vegetables. Have a lot. It's 2 words. I swear. Okay. So they contain starch for energy storage. Other organelles perform specialized functions. Peroxisomes and glyoxysomes, which are only in plants. They're another one of those special organelles that are only in plants. So peroxisomes collect and break down toxic byproducts of metabolism. We went over this on the diagram, such as H2O2. So peroxisomes break down peroxide. So that's a good way to remember that, using specialized enzymes. Glyoxysomes, which are only in plants, lipids are converted to carbohydrates for growth. So they take lipids, which animals and plants both need lipids for their cell membranes, and they convert them to carbohydrates, which they use in the cellulose for their cell walls and things like that. So they take the fatty acids, and they convert them into carbohydrates. Vacuoles, mainly in plants and fungi, have several functions. Storage of waste products and toxic compounds. Some may deter herbivores. So that's like nicotine and THC. That's supposed to deter, but humans apparently like them. Tea leaves too. We somehow like all these tastes that are supposed to deter herbivores. The structure for plant cells, water enters the vacuole by osmosis, creating turgor pressure. So since plant cells have a cell wall, they need that central vacuole to keep everything nice and stiff so that a tree is nice and it stands up rather than wilting over like that. This is a happy tree, lots of water. This is a sad tree, not much water. Or broccoli, or a celery stick, whatever you want that to be, that's what happens. Reproduction. Vacuoles in flowers and fruits contain pigments whose colors attract pollinators and aid seed dispersal. So that's a good thing for plastids to do. And then catabolism is the digestive enzymes in seed vacuoles hydrolyze stored food for early growth. So that's just another reason for vacuoles.